I am going to talk about biological classification of microorganisms. Microorganisms are nothing but organisms invisible to naked eye and are visible under a microscope. These include viruses, bacteria, algae, fungi, and protozoa. Classification of microorganisms is called microbial taxonomy. Microorganisms can be classified in the following ways: based on plants or animals, based on cellularity, based on the type of nuclear materials, five kingdom concept, eight kingdom concept, three domain system. So, classification of microorganisms based on plants or animals. Aristotle in 4th century he developed two kingdom concept. According to this two kingdom concept, living organisms can be divided into two kingdoms like Plantae and Animalia. Plantae includes algae, fungi and bacteria. Animalia includes protozoa. Classification based on cellularity. According to cellularity, microorganisms can be classified into non-cellular, unicellular and multicellular. Non-cellular means no cells. For example, viruses. They are living chemicals. Unicellular means single cells. For example, protozoa, bacteria, some algae and some fungi. Multicellular means fungi, algae, plants, animals, etc. Classification based on the nuclear materials. Based on the nuclear materials, microorganisms can be classified into prokaryotes, eukaryotes. So prokaryotes means those organisms they don't have nuclear membrane, proper nuclear membrane, and their nuclear materials are remain scattered in the cytoplasm. So this type of nucleus is called nucleoid or incipient nucleus. They contain 70s ribosome. Their chromosome is circular in shape. In this, cell organelles are absent. Example, bacteria. In eukaryotes, the organisms contain true nucleus. Their nuclear materials are surrounded by nuclear membrane. They have 80s ribosome. Their chromosome is not circular in shape. In this, cell organelles are present. For example, protozoa, algae, fungi, and all animals. Five kingdom concept. Whitaker in the year 1969 proposed five kingdom concept for the classification of microbes according to mode of nutrition and cellular organization. Based on five kingdom concept, microorganisms can be classified into five types: Munera, Protista, fungi, Plantae, and Animalia. So, Monera includes prokaryotes. They lack digestive mode of nutrition. Example, bacteria, cyanobacteria. Protista includes unicellular microbes like protozoa, unicellular algae, unicellular fungi, and they are autotrophic, holozoic, and heterotrophic fungi. They are all eukaryotic fungi. They show absorptive nutrition and filamentous structure. Plantae. They are eukaryotic multicellular plants. They show photosynthetic mode of nutrition. Animalia includes eukaryotic multicellular animals. They show ingestive mode of nutrition. Eight kingdom concept. Cavalier and Smith they proposed eight kingdom concept. According to this concept, microbes can be divided into two empires having eight kingdoms. In these two empires includes bacteria and eukaryota. Empire bacteria again divided into two kingdoms, that is eubacteria and archaebacteria. Eubacteria means true bacteria, archaebacteria means filamentous bacteria or ancient bacteria. Eukaryota includes all eukaryotic organisms. Eukaryota includes six kingdoms: Archozoa, Protozoa, Chromista, Plantae, fungi, and Animalia. Archozoa it includes microscopic animals feeding on archaebacteria. Protozoa includes unicellular animals. Chromista includes photosynthetic organisms that contains chromoplast, for example, diatoms, cryptomonads, and brown algae. Plantae includes all photosynthetic plants and algae. Fungi includes eukaryotic multicellular and they shows absorptive mode of nutrition. Animalia includes multicellular animals. They shows ingestive mode of nutrition. Three domain system. Woose, Candler and Wheelis in the year 1990 they proposed three domain system based on comparative analysis of ribosomal RNAs. In this phylogenetic system, there are three domains. One is bacteria and another one is archaea, third one is eukaryota. So in this bacteria, that includes unicellular filamentous prokaryotes that having diacylglycerol diester and eukaryotic type of rRNAs in the cells. Archaea means ancient bacteria. They live in extreme conditions like hot, cold, like salt, etc. Eukaryota includes glycerol, fatty acyl diester and eukaryotic rRNAs. Monera. Monera includes all prokaryotic organisms. Pro means primitive, karyon means nucleus. In these organisms, there is nucleus is without nuclear membrane. That is, no membrane-bound structures are there, or membrane-bound structures or cell organelles. And Monera includes Archaebacteria, Eubacteria, Cyanobacteria, and Mycoplasma. Archaebacteria means it is an intermediate between prokaryotes and eukaryotes. It is the missing link. And in Archaebacteria, cell wall lacks peptidoglycan. It lives in extreme environments like salty lakes and volcanic springs. It includes three groups: halophiles, thermoacidophiles, and methanogens. Halophiles mean salt lovers, and methanogens means methane producers. Eubacteria means true bacteria, that is, their cell wall is made up of peptidoglycan. They are present day bacteria or modern or advanced bacteria. Cyanobacteria, that includes blue-green algae or blue-green bacteria. They are the connecting link between bacteria and green plants. They are prokaryotes. They are gram-negative bacteria. They are photoautotrophs and they have heterocysts. Mycoplasma or PPLO, that is pleuropneumonia-like organisms. They are prokaryotes without cell wall. They are gram-negative organisms. Shapes of bacteria. So bacteria may occur in various shapes: circular, oval, comma, spiral and mycelial structure. Circular means that is called a coccus. If one individual coccus occur means that is called a micrococcus. If two two coccus means that is diplococcus. If there is a chain of coccus means that is called a streptococcus. If there is a cluster of coccus means that is called staphylococcus. Same like that bacilli also. Diplobacilli, streptobacilli and staphylobacilli. Comma shape is called vibrio. Spiral shape is called spirillum. Mycelia, filamentous bacteria. So this picture shows the various shapes of bacteria: coccus, bacillus, vibrio, spirillum, spirochete. So this shows the cocci. This is monococcus, this is diplococcus, this is tetracoccus. Tetra means 4, diplo means 2, mono means single, strepto means chain of coccus, staphylo means cluster. So this is bacilli, not common bacilli, diplobacillus, streptobacillus, staphylobacillus. So bacteria is either motile or non-motile. They are with or without flagellum. If flagellum is present means that is called a trichus. If it is absent that is called atrichus. If in bacterium there is one flagellum occurs means that is called monotrichus. In single flagellum that occurs in both ends that is called amphitrichus. Many flagella occurs at one end that is called cephalotrichus. There is a cluster of flagella occurs in two ends that is called lophotrichus. Flagellum occurs all around that is called peritrichus. If the flagella is present only in bacilli and spiral bacteria, it is absent in cocci. So this picture shows the type of flagellation in bacteria: atrachus, monotrachus, lophotrachus, amphitrachus and peritrachus. So in flagellum there is a basal body, hook and flagellum. So the basal body having rings, in the 4 rings in gram-negative bacteria and 2 rings in gram-positive bacteria. There are 4 types of rings: M ring, S ring, P ring and L ring. M ring is called membrane ring, S ring is called supermembrane ring, P ring is called peptidoglycan ring, L ring is called lipopolysaccharide ring. Gram staining. Hans Christian Gram in 1884. It is a differential stain which distinguishes between gram-positive and gram-negative bacteria depending on the difference in the cell wall structure. So these are the stains used in gram staining. That is, one is crystal violet, gram iodine, decolorizer, gram safranin. So the crystal violet acts as a primary stain. It is violet colored. It stains all the microorganisms. Gram iodine acts as a modern stain. So it forms crystal violet iodine complexes. Decolorizer, that is acetone plus methanol. This removes crystal violet iodine complex from thin peptidoglycan layers. It dissolves outer layer of gram-negative organism. Gram safranin, it acts as a counter stain. It is red colored. This stains thin-walled gram-negative organism. So this picture shows various staining of gram staining: crystal violet, grams iodine, grams decolorizer, safranin. This picture shows staining of gram positive and gram negative bacteria. Gram positive bacteria is purple in color, gram negative bacteria is pink in color. So this picture shows the fixation staining of gram positive and gram negative bacteria. Both procedures are same, some with some modifications. First step is fixation. First we should fix the bacteria, either gram positive or gram negative. Then we should add crystal violet. Then we should add iodine. Then we should decolorize the slide. After decolorization, the some bacteria becomes purple in color. So this purple color bacteria is called gram-positive bacteria, and some bacteria lost the purple color. So this bacteria should again counterstain with safranin. After it will change into pink color. This pink color bacteria is called gram-negative bacteria. So this picture shows the difference in cell wall structure of gram positive and gram negative bacteria. So in case of gram positive bacteria, there will be a peptidoglycan layer. This is the outer layer. This peptidoglycan layer is very thick when compared to gram negative bacteria. Whereas in gram negative bacteria, apart from peptidoglycan, there is one more layer that is called outer membrane. This membrane is made up of lipoproteins and polysaccharide, and the thickness of peptidoglycan is very less when compared to gram positive bacteria. So this picture also shows the cell wall of gram positive and gram negative bacteria. Why gram positive bacteria resist decolorization by alcohol or acetone mixture, but the gram negative ones cannot? Because the cell wall of gram negative bacteria has high lipid content and low peptidoglycan content. So when treated with acetone or alcohol mixture, dissolves their lipids, creating pores in the cell wall through which crystal violet dye will diffuse and gram negative bacteria become unstained. On the other hand, gram positive bacteria has high peptidoglycan content and low lipid content. So when treated with a decolorizer, it will dehydrate the cell wall, causing it to shrink and the pores to close, and thus the diffusion of the dye will be blocked. principle. Gram-positive bacteria, when treated with a basic dye such as crystal violet, then with iodine, and then subjected to a subsequent treatment with a decolorizing agent such as alcohol or acetone mixture, remain stained with the basic dye, and the decolorizer doesn't remove the dye. However, gram-negative bacteria, when treated similarly, will be readily decolorized with the alcohol acetone mixture and become invisible. Thus, to render the unstained gram-negative bacteria visible, a counter stain is applied. Gram-positive and gram-negative bacteria shows difference in their cell wall. In gram-positive bacteria, cell wall is made up of peptidoglycan that is rich in teichoic acid, and the cell wall is thicker. In case of gram-negative bacteria, cell wall is also made up of peptidoglycan, but there is no teichoic acid and the cell wall is thinner. One more layer, that is lipoproteins or polysaccharide layer. Gram-positive bacteria have a thick peptidoglycan layer and no outer lipid membrane. Gram-negative bacteria have a thin peptidoglycan layer and have an outer lipid membrane. This is the staining procedure. So first we should heat fix the bacteria in a slide. Then we should treat it with crystal violet approximately 30 seconds to 1 minute. Then wash the slide. Then add grams iodine on the slide for 30 seconds to 1 minute. Then all the bacteria appears in purple. Then wash it with acetone or alcohol, that is 90%. In this, some bacteria appears purple, that is called gram-positive bacteria. Other bacteria lost color. So this bacteria should again counter-stained with safranin. Then that appears pink color. This is called gram-negative bacteria. This is the same procedure. So this picture shows gram-positive and gram-negative bacteria. Gram-positive appears purple color, gram-negative appears in pink color. Thank you.